My name is Meg DeAngelis, and I'm here today to talk about research in my laboratory. But first, I'd like to introduce myself. I'm an endowed chair and professor at the University of Buffalo at the Jacobs School of Medicine in Buffalo, New York, of course. And my research looks into the genomic and molecular underpinnings of age-related macular degeneration. And one of the most important aspects of my lab is to train the next generation of scientists and clinician scientists to go on and further this area of research. That being said, because of the generous funding of the macular degeneration, we're able to do a lot of work that is based on our donor eye repository. And this is very important because there are no animal models that recapitulate age-related macular degeneration. And that is because animal models do not have a macula. The macula is very unique to humans, and that is where age-related macular degeneration occurs. That is very important to know because we do not know why it affects the macula. And that is very important because in all the diseases of aging, that's what gets attacked in this disease. So our lab is focused on trying to understand that. And we use a systems biology approach. And that means using more than one approach, and it's a way to internally validate what you're doing. So we look at it from a gene expression level, a DNA level, microRNAs, and using several methods to try to understand what's going on. Because certainly this disease runs in my family, I don't want to get the disease. So that's one of the impetuses that drives me to study this disease. So one thing we've been doing, and with the help of the Macular Degeneration Foundation, and since coming to Buffalo, with the help of the Lions Eye Bank in Tampa, Florida, is we've been able to expand to other populations of different ethnicities and races to understand why certain populations suffer from this condition more. And to understand genetics, epigenetics, and gene expression and how they all work together at the single cell level. And one of the greatest things I think we did this year, in the past year, is understanding how the environment and the genome come together to influence disease, particularly in the dry form of the disease. In the intermediate form, and in the late stage, atrophic form or geographic atrophy. And by understanding that and looking at more than one aspect or one gene, it's possible to understand how we can look at more than one target at a time or modulate more than one target at a time. And that may help us to understand why we have to maybe think about looking at modifiers of disease. And this is not a new idea. This is something that's been used in cancer for a long time, particularly in lung cancer. And it's something that we've talked about within our own work in age-related macular degeneration now for 15 years. So this is just really coming full circle again, but with the donor eye bank. And also looking at biomarkers of disease to try to pick who in those with the intermediate form of disease are going to progress to the more severe form of the disease. And looking at different pathways and aspects of that, all while training the next cadre of students, whether they be medical students, graduate students, or undergraduate students, or even residents. And I think that's what the greatest passion and joy is, anyways, for me, with working with the students, is working with the students and having them present that work and author it.
With your work being in genetics, how are you using the eye bank and eye tissues to look at macular degeneration in the macular tissue in relation to other parts of the eye?
So part of the thing that we do, and this has to really do with the working with really extraordinary collaborators like Dr. Leah Owen, Dr. Ivana Kim. I really want to bring attention to my clinical collaborators because working together, we have developed a really rigorous post-mortem phenotyping protocol where we take every eye, and the beauty of the donor eye bank is using fresh tissue so that you can separate true disease pathophysiology from eyes that go through post-mortem changes, death changes. And tissue, any type of tissue, that once the death is declared, tissue is going to undergo change. And that tissue really needs to be processed from the time of death to processing in under 6 hours. And working particularly too now with the Lions Eye Bank in Tampa, Florida, we've gotten that down to 3.5 hours. We position the eye as if the patient is sitting in the chair, so it will have the greatest translational value. And we use the same type of multimodal imaging that you would if the patient is sitting in the chair. And those images are then transferred to at least 6 specialists that we all work closely together for them to phenotype, for lack of a better word, blindly or independently. And there's not consensus in the grading. And we base it on the ARIS, the Age-Related Eye Disease Simplified Study Scale that was developed by Emily Chu and Rick Ferris. And a shout out to Emily Chu for winning the Proctor Award for that, very deserving. Based on that, but we've added optical coherence tomography, another type of imaging, but we use that to rule in and out artifact. Because certainly when the eye's out of the body, there's things that can look like they're drusen or a tear that might look like it's a neovascular membrane. It could be mistaken for wet AMD, when in fact it's not wet AMD. So it really helps to separate those postmortem changes. And what we have found is that 30% of the population over the age of 60 is not normal. And that's really important because that's a lot of people. Now, they obviously were not blind from their vision, but they weren't normal either. And I think that's what helps us, is that rigorous phenotype to really tease out what is truly associated with disease from what's truly not. And what we found out with also the rigorous dissection protocol and separating that macula from the periphery, as we have found in our recent cell genomics paper that we collaborated with Genentech on, and I'd like to give a shout out to Luz Ozorko and Mei Chen, who else were senior corresponding authors on there, that we were able to show that there was differential expression between the macula and periphery. And had we only studied the periphery, we would not have picked up those changes. And a lot of papers only studied peripheral tissue, or a lot of studies only study the periphery. And we also focused on the retinal pigment epithelium. And a lot of studies only focus, say, on the neural retina. And there's different beliefs about, does the disease start in the neural retina or does it start in the retinal pigment epithelium? And it's really important to understand the type of tissue as to where the disease starts. And we're strong believers, as are others, about understanding what type of tissue and to study the affected tissue and to study aged matched controls and the same ethnicity. And we'd like to study other ethnicities. And I think it's very important. And I think that's what's led to some of these discoveries we would now like to test out and test therapeutically. And then once you discover things this rigorously, you can then go into animal models. Their animal models certainly serve a purpose, but you've got to start somewhere and then work that way.
Can you talk about the large genome-wide associations that have been done?
Okay, so many years there's been many genome-wide association studies done, but we have to remember those have been done on peripheral blood. We don't know as yet, is age-related macular degeneration a systemic disease or is it a localized disease? Is it only a disease of the retina pigment epithelium or of the retina? Or are there aspects to the disease that have components of both? There's both localized aspects and systemic aspects. And so it's important to understand this and look at gene expression also. And we have shown that there's certainly differences, particularly in our recent cells paper, of which Trifa Swami, a graduate student in my Ph.D. graduate student in my lab, and Charlie Zhang were both co-first authors, where we show there's distinct differences between the macula of the retinal pigment epithelium and distinct differences in the retina, the neural retina macula, and there's very little overlap between the two. So that's really important to understand. So that tells you there's very different transcriptomic architecture between the two. And then we have to think about the way those genes are expressed. And genes, if I have to remind myself about this, genes in and of themselves are expressed many different ways. One gene can be expressed 4 or 5 different ways, and that's called different transcripts. And 95% of our genes have very different transcripts. And we found that the differential transcripts are different between the macula retina and the macula retina pigment epithelium, and how that comes together with gene expression, with how the genes are expressed in general, and how we can further target may help too to drive therapeutics, like getting to the exact transcript and how that overlaps. But it was nice to show overlap between the different, because it was 2 different sets of donor eyes that we used. And it was nice to show that we were able to validate our findings from one paper to the next. And that gives you greater strength and confidence in your data to move forward to the next step when you're making models to test therapeutics.
Last year, Dr. Zhang presented on a paper that described how some genes are expressed in protective pathways. Can you touch on that a little?
So another thing that I find very important, that it is a big area of investigation in our laboratory, and I'm sure as well as other laboratories with age-related macular degeneration, is protection. Some people might refer to it as neuroprotection. You'll hear that term used a lot in several diseases, whether it be age-related macular degeneration, glaucoma, or any type of neurodegeneration. And when we always think about increased risk, but what truly decreases risk? So just because you have absence of an increased risk factor doesn't mean you're protected from something. but really truly having a protective factor, an associated protective factor. And what we like to see are factors when you see something that's associated with a decreased risk. Oh, wow, that we see that people who are controls, we wonder what's driving that expression up in those controls. Why are those people protected? Because we'll see certain individuals that they might have all these known environmental risk factors. They might be obese, particularly the ones that might be very obese, heavy smokers, heavy drinkers. Those are very rare, but they get no disease whatsoever. What's protecting them? Or you'll just see people that follow the right kind of diet or slim and they develop no disease. But what's interesting is to see folks that maybe have an eye exam of a 40-year-old and they're 88 years old and they have no pathology, not even tiny little drusen that you see in the periphery. What's keeping them protected?
Is there anything you'd like to close with?
Is thank my extraordinary funders, in particular the Macular Degeneration Foundation. And I want to thank my wonderful mentees, Trifa and Charlie. And I want to thank my extraordinary collaborators, Leah Owen and Ivana Kim, for this. None of this work would be possible without them. And really, we want to knock this disease on its head.
Thank you for sharing the progress you're making in your lab. We are excited to be a part of your journey and following your work through the years.